It's Biomed Cafe menu. Histones will be presented today. We will talk about nucleosome, histones structure, histone function, and quiz game. I'm Dr. Biomed, cellular and molecular biologist. Hi everybody, welcome to the Biomed Cafe channel. In this channel, complex biological and medical contents are presented in a simple, classified, and summarized form. Histone discovery. They saw something others didn't see. Histones were discovered in 1884 by Albert Cossell. In the early 1960s, James F. began a study of these proteins that were known to be tightly associated with the DNA in the nucleus of higher organisms. During the 1980s, Michael Gustine demonstrated that histones replaced transcription in vivo, leading to the idea of the nucleosome as a general gene repressor. Nucleosome. A nucleosome is a basic unit of DNA chromatin in eukaryotic organisms. The arrangement of the nucleosomes is similar to that of beads on a string. Nucleosomes formation. The nucleosome is made up of a negatively charged DNA sequence of 146 base pairs or 1.65 loops of DNA wrapped around a core of 8 positively charged histone proteins or histone octamer. Nucleosomes are the fundamental building block of DNA packaging. Nucleosome function. They are as a scaffold for the creation of higher order chromatin structure. They are a layer of regulatory control over gene expression. Difference between chromatin and nucleosomes. Chromatin refers to the DNA wrapped in histones as a whole, but nucleosome refers to the fundamental repeating structural unit of chromatin, the nucleosome core particle or NCP. It is made up of about 146 base pairs of DNA wrapped in 1.65 left-handed superhelical loops around the histone octamer. It is formed of 2 H2A, H2B dimers and 2 H3, H4 dimers. Adjacent nucleosomes are linked together by a free DNA stretch known as linker DNA, which varies from 10 to 80 base pair in length depending on a species and tissue type. The whole structure produces a cylinder with a diameter of 11 nanometers and a height of 5.5 nanometers. Histones. Histones are abundant in lysine and arginine. They are found in eukaryotic cell nucleus and most archaeal phyla. Histones supply energy and a structural surface for the DNA that wrapped around them. Histone major families. Histones can be divided into two families: core or nucleosomal histones and linker histones. Core or nucleosomal histones. These histones can be divided into 4 types: H2A, H2B, H3, and H4. The core histones exist as dimers, H2A and H2B. They combine to form dimers that are then placed onto nucleosomes by the nucleosome assembly protein 1 or NAP1, which also aids in nucleosome sliding. H3 and H4. They are put together by the chromatin assembly factor 1 or CAF1 complex, which is made up of 3 subunits. Core histones function, allowing for interaction between distinct dimers, particularly in a head-tail fashion. Linker histones. H1. The linker histone H1 binds the nucleosome as the entry and exit sites of the DNA. Function of linker histones. Locking the DNA into the place, allowing the formation of higher order structure. Histone function. Number 1. Compacting DNA strands. Histones act as spools around which DNA winds. This enables the compaction necessary to feed the large genomes of eukaryotes inside cell nucleus. The compacted molecule is 40,000 times shorter than an unpacked molecule. Number 2. Chromatin regulation. Histones play important roles in gene regulation and DNA replication. Histones undergo post-translational modifications that alter their interaction with DNA and nuclear proteins. These modifications are methylation, acetylation, phosphorylation, and ubiquitination. Number 3. Damage protection. Histones prevent DNA from becoming tangled and protect it from DNA damage. Key points of histone. Each nucleosome is made up of around 2 rounds of DNA wrapped around a group of 8 proteins called histones, known as a histone octamer. Histones have 2 major families, core histones and linker histones. Thank you for watching. Please subscribe to this channel, and if you like the contents of this video, please like it. Now it's time to the quiz game. I've put the link to the quiz game in the description of this video. Download it for free and enjoy it. Until the next video, bye!